Chapter 19 is about nucleic acid, and we're going to talk about the DNA portion of nucleic acid, RNA portion of nucleic acid, and then how RNA gets transformed into protein. I'm going to start with this big picture idea of DNA and protein and nucleic acids and where it is. So this is down on the bottom is just an image of a cell. And you can see that we have different components of the cell. There are different things in the cell. We're going to just focus on the nucleus and the DNA inside. And the DNA is composed of these chromosomes, and you get one of these portions of your chromosome from your mom and one from your dad. And within those are these different complex groups of proteins and DNA that are glued together through these nucleosomes, and then we also can get smaller and smaller and kind of dig into that structure more to see that we have these base pairs, and this is what we would call our DNA. So here's just an image of the different chromosomes in humans. They're all different and unique, and this is a karyotyping technique. So we can actually get these things to different chromosomes will take up different dyes depending on how they're manufactured, and you can kind of see then they can be organized into this different kind of organization. And this is how we karyotype cells to say if they're normal cell types in humans, for example. So these are all DNA labeling, and they're hybridizing different components of the DNA within these different types of chromosomes. Sometimes these are some of the things that are done to look at cells for children. Sometimes these will be screens that are done on some of the cells in a developing human or embryo. And this is the trisomy, where you can have different things that are multiplied or these chromosomes when there's an abnormal duplication of them. So you can get different syndromes. For example, would be Down syndrome for chromosome 21 duplication. So just these are typically just get one from each parent, but sometimes that process can go awry. Chromosomal mutations, this is kind of part of natural progression as evolution is happening, where some things will get separated apart. So this is a Chinese muntjac. It's a type of kind of deer, and an Indian muntjac. And they look very similar, and they have actually very similar overall genes, but you can see that the Chinese muntjac versus the Indian muntjac has a lot more chromosomes. So different animals will have different types and groups of their chromosomes. So even though these are very similar and closely related animals, their chromosomal makeup is very different. The genes are all in here. They're very similar from both sets of animals, but obviously these are going to be separated and different, and now it's going to be really hard or impossible for these to, for example, mate and then have a kind of something that would be able to combine their genetics now because of these differences in their chromosome makeup. So that chromosomal change obviously allows for the species separation. So this is human versus bonobo monkey. So you can see that, we have 22 plus our sex chromosomes. This is obviously a female, doesn't have a male chromosome, but this is a male bonobo monkey, 23 chromosomes. So there's this species separation of the changes in the chromosomal number allow for that separation. Yeast has about 6,000 genes on 16 chromosomes, and there are going to be a lot of different packed information in here. So genes are the things that we're going to express as proteins later on, typically. And there's a lot of information packed into these strands of chromosomes. So you can see these little pink lines. Those are encoding for the proteins that yeast cell is going to make. So the central dogma of the cell biology and genetics is basically for every gene that we have, we're going to make different RNA molecules, and then each RNA molecule is going to make a different protein. So when we're looking back at this slide, this is just 0.5% of the yeast genome, and all of these little pink things are going to be different genes that are going to be transcribed into different RNAs that are going to be translated into different proteins. So this is kind of the idea. This is the central dogma of genetics. And I'm sure that you've probably maybe seen this before in terms of some type of science class in your past with interphase, metaphase, and these are some of the different processing for cell division, and then the ability for us to separate our genetic material apart. So in interphase, we're kind of duplicating these two different chromosomes, and then we're able to separate those out and to have separate cells. And sometimes there's things that cross over, and we can get that variation within our progeny then in that way. Here's a scanning electron microscope image of an actual chromosome. And you can kind of see that's how we get these little cartoon pictures. They look very similar. Here's one half and the other half, and they're kind of connected in the middle with the centromere. So some of the proteins that might be involved in organizing some of this structure together. All of this genetic information is maintained in our nucleus of our cells. And you can kind of see here, this is again a similar type of staining system that was done when I was showing you just the individual chromosomes. And you can see that these things remain pretty compacted. They're not like spaghetti in a pot. They're still kind of compacted with and organized within the nucleus, and different components are available while others might be more packed away. The other thing to note is that these things, this is supposed to be a DNA helix, a double helix from one side and the other. So there's two groups of DNA that bind through this helix structure, but this blue thing in the back is also protein. So these things aren't just floating around by themselves. They're very associated with a lot of different types of proteins, and one of those proteins is called a histone, and they help to wrap up and organize this DNA into usable structures in our nucleus. Here's an electron micrograph of some more DNA, and you can see that these are different chromatin fibers with the DNA. So with the histones and the protein and the DNA in one, and this is more treated to kind of help to unfold it, to kind of see the beads. So these are the little histone proteins that are associated with the DNA molecule. You can kind of think of the histones as like a little hose reel, because each piece of DNA is wrapped around that, and it just helps to organize this structure, this long amount of information that we're trying to store in our cells. Histones are kind of interesting proteins because they have all these little tails and ability to modify those little tails with methylation, acetylation, phosphorylation that can change how these look and how they interact. So it'll actually change if a group of DNA is more open and able to be expressed in terms of making protein from it, or it can be modified in a way that will kind of silence it and keep it packed up and put away, sort of in, kind of think of it as it's got a tag on it to, put it up in the attic, for example, versus having it out on the lawn open for gene expression. So now that we kind of understand how this DNA is packaged, I want to kind of dig in a little bit more in terms of what the molecular structures are of these things, and kind of get away from some of these more cartoonish kind of things. And again, we're going back to the central dogma where we have this double-stranded piece of DNA, and we're going to, there's a gene that we're going to look at, and that gene we're going to do a transcription process where we're going to change from DNA to an RNA molecule. And then, so this is called transcription. And then from the RNA molecule to protein, we're going to use this as a template to synthesize a protein that's called translation. So we go from, typically we generate our transcript from the antisense strand. So if we go from 5' to 3', and then that 5' end becomes the N-terminal end of the protein versus the C-terminal. 5 to 3, N to C is kind of how I think about it, to kind of keep that track of that. So I kind of use also kind of an analogy in terms of our genes. I think of it as a book in a library, and maybe you need to get some of that information out of the book, so you have something that's stored in a safe location. And the RNA is maybe just a photocopy of that page in the book that you need, and then you use that to build the thing that you're going to build, right? So that's maybe a birdhouse, and the instructions for the birdhouse are in a book in the library. You make a photocopy of the instructions, you take it home, and then you build your functional thing that you want. So these are some of the structures of the nucleotides, and what you'll notice is that there is this compound on the bottom, and this is actually a sugar molecule. You can see there's a similarity with some of the carbohydrates we're looking at, where we have this ring structure with an oxygen in it, and we have hydroxide groups coming off the end. And so that's why we call this is the sugar backbone. And it's also has a phospho group on it, or a phosphorylated group on it. So this is a cytidine monophosphate, or CMP. Usually we use this just kind of the C, the U, the T, the A, and the G as distinguishing what type of nucleotide we're talking about. Now this is all the same for each of these. You'll notice that backbone is similar. The difference between C and U, for example, is this part up here. And these on the top are called pyrimidines because they just have this 6-membered ring structure. And if you go down here to the purines, you'll notice it's a 6-membered ring attached to another 5-membered ring. So these are bigger groups of molecules for the purines versus the pyrimidines. And the difference between C versus U is, for example, at this 4th carbon, there is an amine group and versus a carbonyl group. So there's just a structural functional difference between these two. We also notice there's a double bond between the 4 and 5 nitrogen and the 4 and 5 nitrogen. Here there's no double bond. So there are some slight differences between these, and those are going to change how they interact with other nucleotides. You can see that the thymidine also has a different structure, and we have this methyl group coming off the 3 carbon. And so it's similar to the uridine, but that small change is going to make a difference in what it interacts with. Going down to the purines, we have adenosine monophosphate and guanosine monophosphate. And here we have the difference between the 1 and the 6, right? We have an amine versus a carbonyl group. We have another amine coming off the 2 carbon where we don't have that on the adenosine. So these changes are going to make differences between the different nucleotides and how they interact with each other. So we'll look at that in a moment. One of the ways that you can kind of think about if you ever needed to remember this, for example, between a pyrimidine versus a purine, you think pie as in you're going to eat a piece of pie, and the acronym for the cytosine, uridine, and the thymidine is C, U, and T. So pie cut, you cut a piece of pie. So that's the easy way to kind of remember which one's a pyrimidine versus a purine. And for this one, it's pure as gold. So pure adenosine and guanosine. I think of that kind of pure as gold as just a way to remember between the difference between purine and pyrimidine. These nucleotides are polymerized onto each other, so they're attached to each other through this phosphate group. So you have the sugar is the backbone. That's why it's called the backbone, because it's really, these are similar and consistently the same throughout that molecule. But then you have that nucleotide base part. These are called bases because they're nitrogenous. There's a lot of nitrogen, and nitrogens typically act as a base. So that's where it got its name from. So you can see here's the guanine, the thymine, adenine, and the cytosine. So G, T, A, and C. So that's the distinguishing difference between these, this structure. And obviously you can have different organizations and orientations. As the longer this chain is, you can have different patterns that are arising. And we always read DNA sequences and RNA sequences from the 5' end to the 3' end. And that's also because that's the way it gets read and translated and transcribed. And so we'll talk about that process in a moment here. By convention, it goes, we typically would write it out 5 to 3. I should also note out what this means in terms of the 5' end. How does that get that number, and how do we get a 3 number here? It's because we're looking at the backbone of the sugar. When we numbered sugars, and we talked about this during the carbohydrate section, we number sugars starting on this carbon here. So this is the 1 carbon, the 2 carbon, the 3 carbon, the 4 carbon, the 5 carbon. So we have 1, 2, 3, 4, and 5. So this carbon is the 5' carbon. And so this is the 5' end because it is on the end, where the 3 carbon isn't in the end, it's in the middle, right? It's processing through here. And if we look down at this cytosine backbone, 1, 2, 3, this is the end of that chain, and that's why we get that 3' end. So this is, we're talking about the carbon, and it's based off of the sugar molecule or the carbohydrate molecule that's part of that backbone structure. And that's how we get that 5' versus 3' nomenclature. So when we're looking at DNA versus RNA, the backbones are relatively similar, except for for RNA we have the uracil, and then for DNA we'll have the thymine. So these are very similar structures between the two. There's really the only difference is we have a methyl group in the thymidine at this position versus a hydrogen here, and that's really the only difference that distinguishes these two. But these are used exclusively in RNA, and these are used exclusively in DNA. We're not going to mix those up. And otherwise, the other bases are the same between the two. The other difference is we look at the sugar backbone for RNA. We have on that second carbon, we have the hydroxide, and on the second carbon in DNA, we have a hydrogen. So it's deoxy. So we've removed that oxy group. Essentially, that's why we get the deoxyribonucleic acid. So that's the here and versus just ribonucleic acid, because has the hydroxy group in there. Another thing you'll notice in here is that the RNA is here, it appears as single-stranded versus the DNA is double-stranded. And we're going to talk about that in a moment, which of these kind of match up to each other. But they actually will, these bases are going to match up and pair with each other to form this double-stranded helix. But this also, it does actually happen. It doesn't form in a double-stranded helix. It's typically a little more complicated and different. But this is shown over here on the left is how RNA would still interact. So they're going to make these what are called base pairing interactions, and they'll fold and they'll create this different tertiary structure. But we have these base pairs still will happen here. So these are without the backbone being shown, and so we're just looking at the base pair interactions, and these interact and create those helices or those secondary structures. If we're talking about DNA and RNA, we have thymine or uracil will pair with adenine. So it depends on if we're talking about DNA or RNA. So that's the difference between those two essentially here. And here's an example of these molecules interacting through their hydrogen bonding between them. So you can see that we have these two, the T and U with the A. Notice there's 2 hydrogen bonds here. With the cytosine and the guanine, there are actually 3. So these are actually stronger bonds with these. And so we're always looking at, this is where we look at the purine, the pure as gold, and the pyrimidine, cut C, U, and T. Those are going to pair always together, the purines and the pyrimidines together. But notice we're not going to be able to bind a G with a uracil, for example, because we would have this oxygen and hydrogen. They don't match up correctly. They won't bond. They won't make that interaction because these things in the middle, this nitrogen here, this is a nitrogen already has a hydrogen. Those won't interact. We won't be able to hydrogen bond in the middle here. We have an oxygen over here that won't be able to interact because we have an oxygen hydrogen already over here. So these things won't interact in the same way, and they won't be compatible with each other. So we only can bind T's and A's, and we can only bind G's and C's together. So we're not going to mix those up when we're making those secondary structures like we see on this slide here. So it's always the same matching that's going to happen between you can see here G's and C's, A and U if we're talking about RNA versus the TA and the G and C for DNA. So where does this all happen? Where are we kind of duplicating these things and making these things? This is happening, the transcription, so going from DNA to RNA is happening in the nucleus. So this is called transcription, and then we kind of process this a little bit. We grab some stuff out of it, we simplify it a little bit, and then we export it into the cytoplasm. And then in the cytoplasm is where it's made into protein here. So sometimes the amount of transcript we make, so the efficiency of that transcription, we might get more RNA, and typically when we have more RNA, then we'll also get more protein out. So this is a way that we have different amounts of protein coming off for one gene A, for example, versus gene B. There'll be a relative difference in the amount of protein that we get out. So this is a general rule of thumb that we use for understanding gene expression in our cells. So looking at this a little closer, we're going to look at the transcription. So we have the purine and the pyrimidine we're going to attach these different ones. We'll just say this is T and this is GC, for example, between these. So you notice that it's always blue and yellow, and then the red and green are always matched up here. So we're going to make during transcription, so while we're kind of taking this from the DNA version into our photocopy version of the RNA, we're going to use this as the template strand down here. So you'll notice that this top is going to match up. So this bottom part is the template. So we're going to take that as we're going to use that to create a transcript. So you'll notice that the top here for our RNA is going to match the coding part. So this is the template, and we're going to try to recreate this. So we're going to grab this red one here, we're going to put it here, we're going to match up the blue here, we're going to match up the yellow with the blue here. So the template is what we're reading, and then we're matching up the complementary base pair on the other side. So you'll notice that this is kind of the same coloring on the top to get our new transcript flavors, for example. So we also always go from 5 to 3' because that's how it is done. So when we're doing this process, we start on the 3' end of the template strand and we synthesize the 5' end first. So we make 5' and then we go, and then the 3' end gets made last. So that's just the process that we know. We're not going to get into the excruciating details. I'll show you a moment here. But this is always the same way, that it doesn't sometimes go 5' to 3'. It's always, it doesn't read the template strand from the other side ever. It's always the same process where the 5' end gets created first. This is done by a protein called the RNA polymerase. And you can see that 5' end is the newly transcribed RNA transcript, and it kind of rolls through, and you can see the arrow pointing to the right way here. It's gonna kind of bring in incoming nucleotide triphosphates, and it's gonna glue those on and create that template or that strand from the template. Here's some more micron electroscopy, just showing you that we have all of these different RNA strands coming off of this DNA molecule to just show the complexity of how this happens and how these things are happening simultaneously. So it's not like one polymerase is here and it goes down and makes something. It's a ton of polymerases are all attached to any available DNA, and these RNA transcripts are being processed as well as they're coming off of that DNA molecule. There's a lot of information that goes into this process, and we're only doing this in one chapter pretty quickly, but there are a lot of things that are signals that are incorporated into DNA that tell the polymerase where to start and where to stop and how to get recruited to the gene of interest. So there's a lot of information that's put into the DNA sequence that brings other proteins to those spaces and allows them to interact for this transcription processing. This is a little more complex view of this process in eukaryotes or humans, for example. It's pretty complex. There are a lot of little components that come in and bind to different parts of the DNA. You have different proteins that will help to stretch and bend the DNA so that things can get in and start to break this apart and melt it, basically, so that we can start to incorporate and start to transcribe this into the RNA, basically copy part of it. We have to unwind all of this DNA. So there's a lot of different things that are involved in this process, and that's important for getting into the DNA. And there are also processes that happen on the RNA. So we have this initiation of transcription. The RNA gets capped, it gets spliced, it gets polyadenylated. So there are other processing steps that happen before it can get made into protein. But we're not going to go into the excruciating details. I just wanted to let you know that there are other processes that happen in case you want to look more into it or are interested. These processing events are really important to help protect the RNA within our cells to make sure that it gets to the right location and that it gets processed correctly so that it can be made into protein. So if any of these things don't happen, then we don't get the protein out of it. So it's an important step, and it protects some of our cells also from some of the invasion of, for example, RNA-based viruses that might try to kind of hijack our translation system. So these processes are part of that check and balance before we process those RNA signals. Just a little slide note, splicing is pretty interesting because it allows for a lot of variation in our genes. You can kind of see here that if we have a eukaryotic gene, that would be human gene, for example, we have coding regions that are in red, which means that's going to be made into the protein part. And then we have non-coding regions that are called introns. And these get spliced out. And you might think, well, that's kind of dumb. Why are we spending all of this energy putting these introns in if we don't actually need them? Well, there are a lot of interesting ways and regions that we can actually splice these out in different ways. So this is a complex view of these are all the exons for variations of genes that when we make antibodies in our body, this is how we make different antibodies. Or if you get a vaccination, this is the process that your immune cells are going through to create different types of proteins that will bind to various types of viruses or bacteria. And it's through differential gene splicing or that recombination that's kind of the same kind of idea. So we can actually make different genes or different proteins when we have this alternative splicing, which is interesting to do because we can, for example, if we want to take 1, 2, and 3, you can get splice in all of those, but you can splice out certain ones. Maybe you want to splice out exon 1 and just leave 2 and 3 versus we want to splice out number 3 and just get 1 and 2. So there are different combinations of proteins and things that we can then make that might have different functions later on. So this is a nice little simple figure kind of showing you that there are all these processing events, and they essentially give you little tickets to exit out of the nuclear envelope to get translated into protein. So if you're missing any of these little tickets that say that you've been capped, you've been spliced, and then you polyadenylated, if you're missing any of those tickets, you're not able to get out and be translated. And this is for eukaryotes, which have nucleus like humans, yeast even, but not prokaryotes. But bacteria, for example, don't have that processing. They're just kind of more simplified programs for this type of creating proteins from our DNA. All right, so how do we make protein from nucleotides? So there are these codes, right? The G, C, and A's. We get them in groups of 3. And then those are read in groups of 3. And each group of 3 was going to signify to the cell that we want to make a different type of amino acid from that group of 3. So we have alanine, arginine, asparagine, for example. We have these different types of groups that are going to signal and identify different types of proteins or different types of amino acids that we're going to use to generate our protein. These are also called codons, and I actually like this image better. So we start with here. This would be the first codon, the second codon, and the third codon. So we have that circle, secondary circle, tertiary circle. So it's 1, 2, and 3. And then you can see what you'd make: serine, tyrosine, cytosine, for example. These are the different amino acids you'd make from different combinations of those 3 groups. So what peptide would we make from this RNA sequence? So CUC, AGC, GUU, ACC, AU. So let's look at the C and then U, and then we come down to C and we'd make a leucine. Then the next grouping, we start with an A, then we go to G, and then C, so we'd make a serine, and then G, U, and U, valine, A, C, C, threonine. AU, we don't have enough information to know what is going to be made here. We could make a methionine or an isoleucine. It depends on what that next one would be. But you notice there's some redundancy in the code. So if we have a G, G, if there's a mutation here that would change maybe a U to a C or a C to an A, if something were to happen, we'd still get glycine out. We might be able to have small things that change, and it won't necessarily matter for our protein synthesis. All right, so how do we use those 3 base pairs to actually make a protein now? What we do is we actually have what are called tRNA molecules, so transfer RNA molecules, and they're going to base pair with the mRNA. So this is the messenger RNA. This is the RNA that we've generated from our DNA, and this transfer RNA is going to be attached to an amino acid. So here we have the carboxyl end is attached to our transfer RNA, and then the N-terminal end is found here, and then this is the R group. So this is how this would be attached, and these are going to be attached, and they're going to recognize different codons on the mRNA. So the protein that's involved in synthesizing and translating the mRNA into a peptide or a protein is a ribosome. And you can kind of see here we have different sites that are highlighted in different colors of these little ribbon structures here. We have an exit site, we have the P site where the peptide grows, and then we have the A site and where the amino acid comes in. So that's where those names are coming from. So we have mRNA is going to come in here and get bound, and then we're going to have these different components where these things are loaded and transferred. And here's kind of a little cartoon image of what's happening. Methionine is always going to start the translation event, and so we'll have the methionine is going to come in and recognize that mRNA sequence. That's where it's also going to be bound in that P site. Then we have the initiation factor, so we're going to scan through until we hit that correct signal mechanism. So this is the AUG start site, and we're going to bind to that. Then once that is bound and attached, that'll change the structure, and then we're going to have the large ribosomal subunit come and attach and start this process. So here's kind of that process. We'll have the methionine, for example, on the 1, and then we basically continue to synthesize. So as we get something bound in here in that P site, another transfer RNA will come in the A site. You'll notice these are not attached. So what'll then happen is once these are bound and everything's in there, then there'll be a chemical reaction that will break this bond and then create a new bond between 3 and 4. So that's what happens there. So you can kind of see that process. We're going to break a bond and create a new bond. So that's what the enzyme is doing. It's helping process that. So as the next step is the large ribosomal subunit will kind of shift over, and then we will help to push the 3 now the ejected transfer RNA that comes into this group. And so then we have now the 4th transfer RNA attached to the growing peptide, and then an open site here ready to bound a new charged RNA. So here we have the 5th one that is going to be recognizing that antisense code here. Again, this is RNA that's base pairing with these down here, and then we have that new amino acid, and then we'll break this bond, create that bond, and then this will shift over, and then it'll push 4 into the exit site, and then it'll pop out, and then we'll have an open site again for a new transfer RNA to come into. There's a lot of conservation in the ribosomes and how they work and function, and that's why we use a lot of antibiotics. So bacterial antibiotics will be used to target the bacterial ribosomes, and tetracycline, streptomycin, chloramphenicol, cyclohexamide, rapamycin, those all block different parts of this thing here. So we're looking at the blocks the binding of the aminoacyl tRNA to the A site of the ribosome. So if the A site's blocked, we can't synthesize proteins, or the bacteria can't synthesize proteins. So the problem is bacteria will then make mutations and changes to kind of block that from being effective over time. So that's how we get drug resistance, is that they change their the structure of their proteins and can get around it. But these, a lot of times, this is how some of the common antibiotics are created, by understanding the structure of this protein of the ribosome and how we can use that to block protein synthesis in bacteria. So there's enough differences between human ribosomes and bacterial ribosomes where this doesn't affect our ribosomes. So these wouldn't necessarily hurt us. They're just going to hurt the bacteria that they're targeted towards. So overall, what we looked at were we have 4 nucleic acids, we have DNA. That DNA is transcribed into messenger RNA. That messenger RNA is processed and exported out of the nucleus. And we have initiation of translation for the creation of peptides and proteins. So that's overall what I wanted to cover in this chapter. And this, once we get into the last chapter for 20, we're going to be looking at various different proteins that are expressed and things that are going to be used for enzymatic processes that are going to be involved in our metabolism and nutrition and breaking down things, and those are going to be important. And so the processing and the number and the amounts of the enzymes are going to be important for how well these processes are working. And the differences or changes in DNA can change the structure, the function of the enzymes, the effectiveness and the efficiency of those things as well. So this all kind of leads into that process. So now we've kind of looked at some of the macromolecules and how they're organized and arranged, and we understand now from in our cells how we have DNA and how those are used to create different types of proteins. And now we're going to look at how we use those enzymes and proteins for building up some of those structures and break those down for energy.